Hey guys! Today we've got a couple of things to do. We're going to learn about lipids, otherwise known as fats. So, our enduring understanding today, same as it's been for the past couple of days. All living things are composed of four basic categories of macromolecules. You should be well familiar with those categories by now. And we all share the same basic needs for life. So, our essential questions for today: what is the structure or the monomers that compose lipids, and what are the polymers? What do the polymers build? Wait, I can read. What is the structure, monomers of lipids, and what polymers do they build? Got it. What reactions create and break down lipids? What are the functions of triglycerides, phospholipids, and steroids? And how do lipids contribute to the composition of the cell membrane? So while we're going through those essential questions, make sure you can do these things. You should be able to identify and classify different types of lipids, like cholesterol, using structural characteristics, things like the number of fatty acid tails they have, whether they're saturated or unsaturated, whether there's carbohydrates involved. And you should be able to explain the role of phospholipids in cell membranes. So, let's talk about lipids. We're going to start with our first essential question. What is the structure or the monomers of lipids, and what polymers do they build? So, lipid structure. Well, we're really talking about fats, oils, and waxes. So, stuff that you eat like butter, vegetable oil, Crisco, stuff like that. They're used to provide energy for cells. They also help with cellular structure and insulation, like this pleasant sea doggo. He has lots of lipids. Now, lipids and proteins are extremely important when we're talking about the cellular structure. And remember, you're a living thing. You're composed of cells. And remember, don't get confused when I say lipids and proteins. Proteins are a different category, right? But it's both lipids and proteins that compose the cell membrane. We're going to focus today on the lipid components of the cell membrane. There's also cholesterol in the cell membrane, and it gives the cell membrane, it maintains its flexibility. So too much cholesterol and the cells become very rigid and stiff. Not enough cholesterol and the phospholipid membrane could disintegrate. So cholesterol, you hear about cholesterol being bad for your health, but you require some cholesterol, and cholesterol is a lipid. And we'll talk a little bit more about that later. So lipids tend to have two parts. They have a ping pong ball head, it's glycerol, and they have a couple of fatty acid tails. So some lipids have two tails, some lipids have three tails, and we'll talk about the difference between those in a minute. But what you should be aware of is you've got this glycerol ping pong ball head, and you've got these long fatty acid tails. And this is just a generalized diagram. We're going to look at the actual molecular structure in just a few minutes. So here's our hydrophilic head. Hydro means water, and philic means like. So that means the glycerol molecule, it likes water. It's attracted to water, right? So remember, hydro means water. Philic equals like, right? The fatty acid tails, these long things, they are hydrophobic, right? Phobic means afraid of. So hydrophobic means doesn't like water, right? So we've got the head that likes water, and we've got the tails that don't. They do not want to touch water. And that makes sense because think about when you're making spaghetti and you put a little bit of olive oil in the water before you put the pasta in, and the oil just kind of like sits on top and it kind of stays in those little bubbles and it doesn't dissolve and mix with the water very well. That's because these tails are all pointing inwards. They don't want to touch the water. They don't like it. Together, this glycerol head and these fatty acid tails create a lipid. But there's technically two types of monomers here, right? The glycerol head is a monomer, and the two tails are also monomers. So monomer is fatty acid. We should probably put glycerol. And then the polymer, when you put those together, you get lipids. Okay, so you've probably heard about saturated fats and unsaturated fats and trans fats, and there's like a million different kinds of fats, and it's like, I don't get what the big deal is. It's all fat, right? Well, actually, there are some pretty significant differences between the types of fats. So this is the molecular structure of one of those fatty acid tails that we talked about. So if this is the glycerol head, right, the ping pong ball, and here are the tails, right? So we're talking about the tails. These are the tails, right? Now, when we say saturated, that means that all the carbons are single bonded. Remember, we talked about carbon and bonding, and I know some of you asked me for a little bit more information about bonding. So remember, carbon has to have 8 valence electrons in its outer shell. So here's our nucleus, here's our inner shell, there's 2, and then here's its outer shell. It's only got 4 valence electrons in its outer shell. It wants 8. So a good way to satisfy that requirement is to form a covalent bond. And all a covalent bond is it means that this carbon molecule is going to share electrons. That's all it means. So let's take this carbon right here. That carbon has 4 electrons in its outer shell, and it needs 4 more to satisfy that outer shell. This outer shell always wants 8. It's always hungry for 8. Why is it no writing? There we go. Oh, there they are. For 8 electrons, right? But it only has 4. So it's going to form 4 covalent bonds with other molecules around it to be stable. So it's going to share a pair of electrons with this carbon over here. It's going to share a pair of carbons, or a pair of electrons, with that hydrogen. It's going to share a pair of electrons with that hydrogen, and it's going to share a pair of electrons with that hydrogen. So in reality, now that outer shell has 8 because it's sharing those electrons. So whenever you see covalent bond, all a covalent bond is the sharing of electrons. Now, when you're sharing electrons, there's energy stored in that bond. It's energy that holds that sharing relationship together. So the reason we use molecules to store energy is because in every single bond, there's energy stored. And when you break those bonds, that energy is released. And that's the process of cellular respiration. We'll talk more about that later. Okay, so back to saturated fats, right? So here we have this long fatty acid tail, and you can see all the carbons are single bonded. There's no double bonds, right? There's no double lines. There's one here, but we don't care about that. We mean in here, there's no double bonds. I can spell double bonds. This is why I use slides, because I can't spell for crap. Okay. You can think of it as another way. Fatty acid tails are like sponges, and these carbons, they want to bond with as much hydrogen as possible. This is a saturated fat because there is no space. There's no more space for any more hydrogens. It is saturated with hydrogens, right? Saturated means like full, like when you saturate a kitchen sponge, right? So this fatty acid tail is a saturated fatty acid because there is no more space for any possible, you can't fit another one there. Doesn't work, right? All available spaces are taken up by hydrogen. It's a saturated fat. It's a long chain of single carbon-to-carbon bonds, and each carbon is saturated with hydrogen atoms. So you can tell the difference between saturated fats and other kinds of fats because they're solid at room temperature. So butter is solid at room temperature, right? So that's a saturated fat. Lard, which is actually pig fat, is a saturated fat. It's solid at room temperature. And I think I put this on here. Yep, saturated fats, they come from animals for the most part. So if it's a fat that's solid at room temperature, it probably comes from an animal, right? So good stuff. Yummy stuff. the good fats. Well, they're all good fats, but. So butter is made from cow's milk, right? Lard is pig fat. Okay, so now we're going to talk about unsaturated fats. Now, unsaturated fats are different. You can see that there's a double bond here. That means that there are two spaces here where hydrogens don't exist. Because those hydrogens are not there, we call this an unsaturated fat. It's unsaturated because there is not every available space is taken up by hydrogen. And what happened is, instead of these carbons forming bonds with hydrogens, they formed a double bond with each other, right? And you'll also notice that if there's hydrogens that are missing on the same side, it also causes a bend in the tail. So if you see a kink in a tail, in a fatty acid tail, that's a good indication that it's an unsaturated fat or an unsaturated fatty acid. So that long chain of carbon bonds is still there, but there are some double bonds. It can be one, it can be two, it can be more than that. And that causes that chain to be crooked. It's kind of like a bent paperclip. And the reason why is if there were hydrogens here, it would cause the long chain to straighten out. But because they're not here, they're not there to like hold the structure straight, it bends. So you get that crooked tail. We call it unsaturated because there's less available space for hydrogens. There should be some here, but there's not. And you can identify unsaturated fats because they're liquid at room temperature. So things like Crisco, which is vegetable oil, or like Pam that you spray in your pan before you cook stuff, canola oil or olive oil, any kind of oil that you cook with, that's an unsaturated fat. And these come from plants. So coconut oil, peanut oil, sunflower seed oil, sesame oil, all the oils that you cook with. Olive oil comes from trees, right? Okay, so here we have a picture of saturated, right? Here we can see there's hydrogen in all available places versus unsaturated. And we have those missing hydrogens. You'll also notice, and just as an exercise, right? Our unsaturated fat has less hydrogen than our saturated fat. And that makes sense because there's two missing right here, right? So unsaturated fats have less hydrogen than our saturated fats. So our saturated fats, these are saturated. They have more hydrogens, single bonds only, and straight. They're in a straight line. Whereas our unsaturated guys down here, right? These guys have less hydrogen. They have some double bonds. It could actually be a triple bond too. And they're bent. So there's three major differences right there between the two. Remember, our objectives, we talked about being able to identify different types of lipids based on their structure, right? And these are the kinds of things that we're talking about. So whether it's straight or bent, whether it's saturated with hydrogen or not, stuff like that. Oh, I have stuff in here. The double bond reduces the need for more hydrogen atoms to stabilize the molecule, right? So that's all you're saying is that, or all I'm saying is that unsaturated fats have less atoms, but this is still a stable molecule, right? That carbon is still happy. It's still got all those covalent bonds that it needs. It's still got all those electrons. It just borrowed it from its next door neighbor instead of forming a relationship with hydrogen instead. Okay, essential question number 2. What reactions build up and break down lipids? So here's that glycerol ping pong ball head, right? So how does it bond with fatty acids? Here are our fatty acids tail. Well, the answer is the same as it was for carbohydrates. Anytime you're building Anytime you're building, it's dehydration synthesis, right? Anytime you're making a bigger molecule, it's dehydration synthesis. Sometimes it's called condensation. Okay, so here we have one fatty acid tail. Here we have a second fatty acid tail. Here we have a third fatty acid tail, and we have that glycerol head. And hey, look, I have these H's and those O's highlighted. What do you think is going to happen there? Well, when you have a condensation or a dehydration reaction, what's the molecule that always pops out? What do you end up with as a product? You end up with water, right? So it's going to form a water molecule. In fact, it's going to form 3 water molecules. Oh, I don't know why that's all messed up. But yeah, you're supposed to see H2O 3 times. So for each time a tail attaches to that glycerol, you get 1 molecule of water, right? So we had 1 molecule formed here, 1 formed here, and 1 formed here. So in order to put together this triglyceride, and we'll talk about triglycerides in a minute, you've got 3 molecules of water. So let's look at that again without all my scribble and scratchy marks. Okay, so this is the ping pong ball glycerol head right here. Right? So how does glycerol bond with these fatty acids? Now we have 3 tails. We have 1 tail here. We have the second tail here. We have the third tail here. Remember, these heads are hydrophilic. And the tails are hydrophobic, meaning they don't like water. Okay. So how do these bond together to form a triglyceride, which is a type of lipid, a polymer? Dehydration synthesis. These hydroxyl ions are going to bond with this hydrogen, this hydroxyl. This hydrogen is going to come away, and we're going to form water. Okay, now that same process is going to happen 2 more times with this tail and with this tail. So each time a tail bonds to that glycerol head, you're going to form another molecule of water. Okay. So here's that triglyceride again, right? How is this lipid broken down? Well, hydrolysis. Remember, hydro means water and lysis means to break. So by adding water, we can break down these molecules again. That's why you get like a giant Coke with your cheeseburger, because you get thirsty because you need a bunch of water to break down those fats. So here's water, and it's going to split apart that glycerol molecule from that fatty acid tail. Here comes another water molecule, and it's going to break that apart. Here comes another one, and it's going to break that apart. Okay? So it took 3 molecules of water to break apart this one triglyceride. Okay, so that was a number 2. Let's move on to number 3. What are the functions of triglycerides, phospholipids, and steroids? So phospholipid, here you go, is used in cell membranes, and we're going to talk more about that in a few minutes. Its structure is 1 glycerol molecule at the top and 2 fatty acid tails. Now, these can be either saturated or unsaturated. A triglyceride, tri means 3, right, is used to store energy. So not structure, it's used to store energy, right? And its structure is that same glycerol head, but now we have 3 fatty acid tails. And again, those can be saturated or unsaturated fats. So they can be bent or straight, right? So phospholipid, 2 tails, used for cell membranes, right? Triglyceride, 3 tails, and it's used to store energy. Okay. Another type of lipid that you need to know is the entire class of steroids. So when you hear about steroids, you think about like grr, like bodybuilders. You can't see me, but I'm doing like muscle arms right now. Steroids, you think of anabolic steroids where people use them to like bulk up and build more muscle mass. Steroids, really, that's not just what they're for. Steroids are a chemical, it's hundreds of different chemicals that your body produces that can help send signals, right? So they tell your cells what to do. Your cells aren't super smart. They need to be told what to do. Steroids help tell your cells what to do. So one particular steroid that's very useful in your body is cholesterol. And cholesterol is a lipid. It's got a glycerol head, but more importantly, it's got that fatty acid tail. This is that fatty acid tail. That's another way sometimes. So we've been looking at fatty acid tails like this. Right? But sometimes you'll see them drawn as like a kinky line like that, or you'll see them drawn as a series of CH2s because here's a C and it's got a hydrogen on the other side, so that's a CH2. So there are different ways to draw it, but you should be able to recognize this chain of CHs as a fatty acid tail, okay? But what makes this particular lipid different than other types, this particular steroid cholesterol, is that it's also got these funky ring things in here. Now, that ring structure is definitely identifiable as a particular class of macromolecules that we already talked about. That's very clearly a carbohydrate ring, right? So cholesterol is a fatty acid tail with a chain of carbohydrates attached to it. And you can see that on the drawing here, right? So we have a hydroxyl for a head. Here are our carbohydrates, but it's got that fatty acid tail on it. That's cholesterol. And cholesterol is super important in maintaining the cell membrane. So here you go. It's 4 lipids, or lipids with 4 fused carbon rings. So here's the lipid tail. Let me get all my chicken scratch out of here so you can see better. So all these things, the blue ping pong balls with the tails, those are phospholipids. Right? But these things, the yellow things, those are cholesterol. It's got that carbohydrate attached to it, and these guys help maintain the fluidity of a cell membrane. Cell membranes need to be flexible, right? So they can take stuff in and spit stuff out, but at the same time, they also need to like hold together. They can't just like, diffuse and fall apart. So too much cholesterol will make the cell membrane very rigid and stiff, which means that stuff can't diffuse through it, and that's a big problem. But not enough cholesterol can cause problems because your cell membrane will start to spread out and like the cell will pop. Now, you only see this in animals because plants don't need cholesterol because they have cell walls, right? They have cell walls that help their cells maintain structure. So instead of cell walls, animals have cholesterol to help maintain that structure of the cell membrane. So how does fat travel, right? We've been talking about fats, saturated fats and stuff, and this fat apparently flies. But I have a quick video for us to watch that shows exactly how fat travels. So like once you eat fat, like where it goes to and what happens to it. So this is a HHMI lecture. And the guy you're going to see is Dr. Watson, one of the discoverers of DNA, and he's going to eat a cheeseburger. But really why we're watching this video is because we're going to look at how fat travels through the body and how it gets from the food you eat to like the places where it's stored in your body.
And Francis is known for his discovery, along with Jim Watson, of the structure of DNA. And he would occasionally indulge in a double bacon cheeseburger from Jack in the Box, and this ultimate cheeseburger contains more than 1,000 calories, 62% by fat alone. And since the requirement for an average person is only 2,000, this would be more than half your daily requirement, not counting the French fries and the Coke that he actually had on his table with him. And that would take you out for the day, that's for sure. Now, food's a mixture of protein, carbohydrate, and fat. And we need the energy from these things to live on a daily basis, but we may not need all of it. And I'm going to take you on a journey as we take a bite of food and watch as it goes through our bodies from the outside to the inside and what happens to it. And it goes through a series of scripted events. Here's the bite of food that through chewing is broken up, and then through biochemical events begins to be further processed in our bodies. Here it is in our stomach, where it's beginning to break up into its components. The little yellow droplets there are the lipids that we're going to track in more detail. And you'll see that these begin to break up into smaller components through biochemical action. And these will drift from our stomach into the intestine, where absorption of nutrients occurs. And here are the microvilli in the intestine that take the fat from the outside, repackage it on the inside into something called chylomicrons, which travel through the lymphatics, ultimately to the bloodstream, where this packaged fat now is delivered to tissues such as the muscle, where it can be used to burn energy, as is shown here, or the excess that is not burned. Remember the fat equation. The excess will be delivered and stored in adipose tissue. This shows some cells there, and our chylomicron droplet delivering the excess energy where it's going to be stored in fat.
Okay, so that's how fat actually travels through the body. It can be sent to muscles for storage and use, or it can be sent to adipose tissue, which is like fat. So like belly fat or arm fat or whatever, fat. So what happens when you have too much fat in your system? We talked about, or we just saw that video, how that fat can travel through the bloodstream in like small packets. Well, this is an artery, right? It's a generic diagram of an artery, right? And these are RBCs, red blood cells. And these red blood cells are flowing through your arteries normally. Well, cholesterol, if you have too much cholesterol and fat, other types of lipids in your arteries, it can start to build a clog, and it causes kind of like a traffic jam, right? So these red blood cells can't get to where they need to be. Everybody's gotten stuck in traffic on the way to school in the morning. It's a pain in the butt. Same thing can happen in your circulation system. Now, because this blood sits here, it can form a clot. This can also form high blood pressure, and eventually this can result in a heart attack. So if your heart doesn't get enough blood, or if there's a complete blockage here, that can cause a heart attack. So atherosclerosis, say it with me, atherosclerosis, is when you get lipid buildup in the arteries, too much fatty acids in the blood and cholesterol in the blood. And actually, your artery will grow a membrane over the top of it and try and like seal it off. But what happens is you get that very narrowed flow. And it's kind of like when you kink the hose, the garden hose, and you get the pressure, the water pressure increases a whole lot. So that's your blood pressure increases a whole lot because you're cutting off that diameter. So that's less room for blood to flow. And it's not like your body decreases the amount of blood, right? So it's pushing that same amount of blood through a smaller area, and you get increased blood pressure. So your total cholesterol, if you have cholesterol, blood cholesterol that's higher than 240, then that's considered high. 200 to 239 is considered like borderline high. Less than 200 is good. So this, you guys won't have to worry about this until you're older, but the older you get, you do have to worry about it because cholesterol comes from two sources, right? It comes from the food that you eat. But we also produce our own cholesterol. So our bodies create our own cholesterol. So you can control your cholesterol by doing two different things, and you have to do both. One, you have to control your diet so you're not taking in extra right? And two, there's medication that you can take that will help prevent your body from making too much of its own. But you have to do both those things in order to get that number down. Okay, last but not least, how do lipids contribute to the composition of the cell membrane? So these notes look a little bit different. These are the notes that I use with my juniors and seniors, but it's the same information that you need to know. So don't be afraid by the understandings on the side. That's just their, you know, how we talk about enduring understandings and objectives and essential questions. Well, they have understandings and applications and skills, and they're right here on the side. So don't worry about that. And like I said, this is stuff that we've been talking about, so don't panic. So the cell membrane is what separates a cell from the rest of its environment. So if you're a living cell, or if you have a bunch of living cells, each of those cells have membranes. You can think of a cell as like a water balloon, right? And the cell membrane would be like the balloon. Now, the structure of that membrane is called a bilayer because bi means two. And it's two layers of phospholipids. You can see it's one layer with the heads that point out and one layer with the heads that point in. And the tails point inwards. So phospholipids are what we call amphipathic. Amphipathic. So if you're like ambidextrous, it means you're both left-handed and right-handed. Amphipathic, sometimes you'll see it written as ambipathic. So sometimes you'll see amphipathic, sometimes you'll see ambipathic. They mean the same thing. Don't let the word scare you, right? But those words collectively, they mean you have both the hydrophobic tails, right? The tails that don't like water. And you have the hydrophilic heads, which is the glycerol molecule on top. And don't worry, I'm not going to make you ever draw a glycerol molecule. Like for me, you can just draw it as a ping pong ball. That's fine. Okay. So you've got that hydrophilic head, meaning it likes water. Hydrophilic means water loving. And you've got those hydrophobic tails. They fear water. So when you put phospholipids in water, they automatically by themselves form this organized bilayer. That's called the self-organization of chemistry. And that becomes really important when we talk about the origin of life. So when you put certain types of molecules in certain types of conditions, they have a predictable and repeatable pattern of behavior. And that's because of chemical properties like the fact that the ping pong balls, the glycerol heads, are hydrophilic, meaning they like water. They don't mind touching water, but the tails don't. So all the tails point inwards because they don't want to touch the water. They don't like it. So this is how you form that bilayer. And I think I have a video that shows how that actually happens. I do. Look at that. Okay. So in this molecular animation, you can see these gray molecules are water. The green molecules are phospholipids. The green are the tails, and then you can see the kind of beige, and then the red and the blue. Those are the glycerol heads. So it looks like somebody just dropped some lipids, like a little drop of olive oil in water, and let's watch and see what happens to those lipids on their own. So look, all those tails are starting to move, and they're all pointing inwards. Right? They're starting to organize. They don't want to touch the water. They're not about that life, right? They're like, ugh. And you can see those glycerol heads are starting to form or starting to organize, so they're on the outside. Look at that. Look how nice and neat it's forming. And this is all on their own, just from those chemical properties. And it's all because those glycerol heads, they're cool. They like water. That's cool. They're friends, right? And all these tails organize nicely. And you come up with this nice, neat, organized phospholipid bilayer, right? Here's the top layer, and here's the bottom layer. We have our water out here, our glycerol heads that don't mind touching the water, our fatty acid tails that don't want to touch the water, And then we have our bottom layer down here. Here's those fatty acid tails that don't want to touch the water. And here are those glycerol heads that do want to touch the water. And you've effectively created a barrier, right? You've got water over here and water over here and this nice organized barrier in between. And that happened all on its own just because of the chemical properties of those phospholipids. This is why phospholipids are the basis of the cell membrane, because they can perform this capability all on their own based on their chemical properties. So this creates two separate regions, right? We've got that hydrophobic interior, no water, and we've got those hydrophilic exteriors, right? So the glycerol heads, they like the water. So this is a micrograph of an actual cell. And you can see here's the cell membrane. And you can actually even see that bilayer, right? You can see the dark line. Those are the glycerol heads. And you can see this kind of clear line in the middle. Those are the phospholipid tails. And then you can see that dark line again. And those are the phospholipid heads. You can actually physically see this. So the membrane surrounds the contents of the cell, the cytosol, or the cytoplasm, same thing, and it separates it from the rest of the environment. So without the cell membrane, there would be no cell. That's like saying, without the balloon, there would be no water balloon, right? Cell membranes are super important. Okay, so here are some review questions. At this point, you should be able to answer all these questions. Now, I'm not going to make you answer them in a quiz form, but what I do have is a Nearpod for you to do. So once you're done with your notes, you can go into, discard those. Once you're done with your notes, let's see. You can go into the rest of the assignment and complete the Nearpod. Okay. And then make sure you upload pictures of your notes and upload your Nearpod results. And I know it's been kind of boring. We're just doing lectures for the past couple of classes. That'll change. We're going to do an enzyme lab this coming week. It'll be a virtual lab. And then we're going to do an osmosis lab pretty soon after that too. So I would like to do the labs at home so that you guys can actually do them yourselves. So let me know how you feel about that. I'll send out a message and I'd like you to respond and let me know how you feel about collecting some materials at home. And then you can document your lab at home. And I'll do it at home too as well. And then we can compare our results, just because I feel like this is so boring. We don't get to do any cool labs. Like usually my classes are packed full of labs. We're always doing labs, but because I'm home, we can't do that. Well, I don't want to leave this up to like walk you through a lab because that's just going to turn into a complete mess. So pay attention. I'll send out messages about doing labs at home. And if you're interested, we can do that. And if you have trouble getting materials, we can talk about that too. Okay. So watch out for that soon. And I'll talk to you guys later.